Je t'aime.
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 Right, okay. I hope everyone can hear me now. Yes? Okay, good. All right. I love to start that way. I love to start on mute. It just gets everyone like excited and focused and trying to help me. So thank you so much. I can see in the chat that you can hear me. And I am really excited about today's webinar on the ViroTEM system. I want to say welcome everyone and introduce myself. My name is Saba Hussein Gore. I am the head of external engagement and applications manager here at Vironova. I'm in Vironova HQ in Stockholm right now. And I'm very excited about today's webinar on the ViroTEM system. I just want to give you some information. During the presentation, feel free to add your questions or comments to the chat area. After the presentation, I will be looking forward to answering these questions. So, no further ado, let's just get started. So, do this. Oh, hang on. So let's get started. Today I'm going to be talking to you about how combining two really powerful tools and methods can give you rapid and routine analytical insights. And that can be done with the ViroTEM system. So, I will start with giving a bit of an overview. So first, I will give you an introduction to Vironova and looking at the challenges then in process development by visual insights. Then I'll follow up with talking about the ViroTEM system and how combining a powerful TEM hardware and a smart AI software can give you really valuable insights. And then I'll wrap up with a summary. And hopefully this won't take too long. I'm going to try to keep this at a sort of medium level, not too much detail, but also packed with useful information, hopefully. And I'm happy to take the questions after the presentation. If anything is unclear or if you want me to go through anything, I'm more than happy to do that. So let me tell you a little bit about Vironova. So Vironova is a Swedish company based in Stockholm. I'm in Stockholm at the moment. And Vironova focuses on nanoparticle characterization insights. And we've got some application areas, or three main application areas, can be basically classified into gene therapy section, the vaccine section, and drug delivery particles. And our solution in those application areas are all about making the invisible visible, valuable, and secure. And our solutions can be divided up into sort of three main categories. One is the actual hardware, the electron microscopes, the systems. The second one is the software, where we have both GMP compliant and non-GMP compliant software. And we have our services as well. We've got the world's first and only electron microscope GMP certified lab. So with that information, I want to jump straight into the next point, which is really addressing challenges in process development by visual insights. So a lot of you might be very familiar with this, but I thought I will still run through this in case there are some new people in the audience to viral vector process development. And when I think about viral vector process development, I think about troubleshooting and hurdles to overcome. It's a fairly long process with many different steps and stages, going all the way from upstream, where you're growing your cells, or you're expressing your viral vectors, and then you go on to harvest this, you purify your particles, you do your formulate, fill, you preserve and store, and then it goes sort of shipment. So there's a sort of a whole trail in the process development. And during these steps, in each one of these steps, there are challenges. Challenges such as your product aggregating. So you can have broken particles because your upstream operations, scale up or something, are not suitable.
You might have problems in removing host cell debris and impurities like DNA, or even process-related particles like rubber or silicone oil that can come, and you find that you can't get rid of it even during your downstream process purification. And then your particles themselves can lose morphology or integrity due to unsuitable conditions. And in order to overcome these challenges, of course, and to address them, you of course need to monitor your product and monitor how your process affects your product. So questions like, is my sample aggregated? How is scaling up affecting my production? Which expression system is keeping my particles the happiest? What happens if I freeze-thaw my sample multiple times? What happens to the integrity of my particles? Which purification method is better? How can I confirm my analysis with several methods to really give me assurance that the data that I'm reading and what I'm measuring is correct? And also, of course, what do my particles look like? And this is where transmission electron microscopy, which I will call TEM for short from now, this is where TEM really comes into play. Because unlike with light microscopes, TEM allows you to actually visualize these small particles that are in the nanometer range. This is where we have our viruses, and you can see that this is really the sweet spot for electron microscopes. You can finally directly visualize viruses, viral vectors, gene delivery, vaccine particles, and so on. So you can actually visually see them and get some visual insights. But not only looking at them, you can actually answer a lot of questions by doing this visual observation. So what can you see? Well, so you could potentially look at particle size distribution. You can look at purity. You can look at integrity, and you can look at morphology. And I'm saying look at, but actually I'm talking about looking at and analyzing and getting solid quantitative data, statistics, hard facts on your particles. Let me now spend a few slides just giving you some examples of this. So when we talk about particle size distribution, we can use particle size distribution analysis to assess sample purity. Here to your left, you can see two samples with adenoviral vectors where we did a measurement where we are measuring the amount of debris in each of the samples. And you can clearly see by visually looking at it that sample 1 has got more debris than sample 2. And actually sample 1 is less pure than sample 2. Sample 2 is the pure sample. So here is a really good way to visually show this, and also to get the quantitative data out of it, to get statistics and to be able to plot this as, let's say, an area ratio of debris in one sample versus another. So this is a really good purity measurement, measuring those very, very small debris particles that can be quite hard to detect in other analyses because they're just too small. That was quite a general measurement, but you can also look at some specific, you can basically reveal some specific hidden contaminants potentially. So the sample to the right, or the image to the right, is an image of AAVs, adeno-associated vectors, with proteosomes in the sample. And this was difficult to decipher using other analytical methods, but with TEM, we were able to pick out these two separate populations of particles that existed in the sample. And again, here we are plotting it, and we can see very clearly what it is. You can use TEM for purity assessment to give you informed process development decisions. Here we are looking at a choice of two different purification methods. One to the left is the Affinity chromatography versus Sucrose gradient. And you can see that the sample that came out from Affinity chromatography purification have got baculovirus still there. This is these sort of long shapes. And this comes from growing the sample, expressing the viral vectors of interest. And you want to get rid of this. So clearly you can see that Sucrose gradient, where you do not find these baculoviruses, is a much better purification method, at least for the step of removing the baculovirus from the sample. Again, helping you to make informed process development decisions. Particle integrity analysis can give you an idea of your particle or your sample stability. Freeze thawing, time, heating, and looking at what is happening to your particles visually and get that quantitative data out of it as well. The example that I've chosen to show here is looking at changes in a formulation buffer for adenoviral vectors and what happens to the particles. You can see that in the old formulation buffer, the particles were quite happy, they were intact and not clustered together at all. The new formulation buffer, however, made changes to that. The particle integrity was lost. They broke and they clustered and aggregated together, giving you an undesired effect. And it's something that you would want to go back and look and assess and make changes and see, well, at which point does this phenomenon or this effect happen? Was it by the addition of, formulation X or something like that? So a very interesting and useful observation that can be very versatile. Assessing overall sample morphology is also extremely useful for example, looking at batch-to-batch variations for quality control, or maybe investigating a deviation. Here you can try to keep track of if there is any changes in the amount of debris, if you're getting more aggregation, or what's happening to your particle integrity. And broken particles, or in this instance is broken AAV particles, are considered to be an impurity that need to be tracked and monitored. You can also look at specific critical morphological features that are linked with biological activity of the product. Here is where high magnification and a really good electron microscope is very, very useful to really pick out those fine details. We have got in this image influenza VLP. The biological activity is the spikes, the HA spikes. And you can see to the left that before polishing steps, the sample was decorated, or the particles are decorated very nicely with these spikes. However, after the polishing step, you lost these spikes. You can see them in the sample, they're not actually on the particles themselves. It's almost like they've been shaved off the particles, clearly showing that the polishing step is affecting the biological activity of your product, and it's affecting it by basically giving it a great shape, shaving off all of the spikes. And I think to see this visually for science, and just to show this even to someone who's not, used to looking at TEM images, you can clearly see something here is not right, and you can guess sort of what that is. Knowing as well that some of the measurements for, some of these VLPs, for example, could be like an ELISA, where you're using these spikes as an indication of the presence of your particles. And if your spikes are just floating around everywhere, that can give you a bit of a skewed result. So combining this together with a ligand binding assay or with sort of ELISA can really help you to understand what is happening and not sort of lead you potentially down a path with not super accurate results, if I may say so in a slightly informal way. So those are just a few examples of the visual insights that you can obtain with TEM, and there are many, many more. This is really, really versatile, and I'm happy to discuss this with people if you are working on something. If you've got some questions about measuring the purity of particular, or let's say, do you have any residual DNA and so on, we're happy to have a discussion and show you some examples of this. But moving on, you can see that there's a lot to gain from TEM images and analysis on viral vectors. And this is where the ViroTEM sort of came about. So the ViroTEM system is a combination of a really powerful hardware and a great versatile intelligence software. The hardware came from Hitachi High-Tech. This is a partnership with Hitachi High-Tech, and the software from Villenova. And combining them together, we get ViroTEM. The ViroTEM, and let me talk to you a little bit about it. It's a really powerful 120 kV electron microscope system. It can give you some beautiful high-resolution images for optimal visual insights. You can see here some examples of this, the detail in some of these fine features, in this instance of mouse kidney, can give you a sense of how precise and how accurately you can really look at some these, for example, like the HA spikes that I was just talking to you about, looking at some of these finer details that are really, really important for the biological activity of your product. And I know that when I've spoken to some clients that are working with, for example, making modifications on spikes, even like so with COVID, you've got your COVID vaccine, you've got the spikes. It is important to see how these spikes, how their appearance change if you make changes to the sequence of it, or if you're making deletions of loops or modifications. So to be able to look at fine details is extremely important and a very useful thing to have. The good thing is with the ViroTEM, you don't only look at things in fine detail, you can actually have a really broad range of applications, a wide span of nanoparticle analysis. And this is because of the wide acceleration voltage of this. It means you can look at things in fine details, but you can also zoom out quite a lot and get a good sense and get a good overview of your sample. In this image, we've got some thin sections of rat kidney, and you can see the beautiful details that you get here and the overview that is extremely useful in many applications. You can run samples through this system really, really fast. And that is really thanks to this one-touch 3-specimen holder. So you can load 3 grids in one go and really ramping up the speed of which you can obtain your data and analyze the images. So you don't have to take it 1 grid at a time, load 3. And then when you're finished with them, you can take that, you can reuse these grids and you can reuse these, redo the analysis if you want to, or image it at a later time. And then finally, we've got the automated features with VS software. This is extremely user-friendly and fast. And let me tell you why fast is important. Fast is important in TEM analysis because actually the majority of the time that is used up is actually on the data analysis itself. So having a smart software really helps you to speed up the time to that insight. And this is exactly what VS does together with this great 120 kV hardware. The image that you're taking get processed really fast, saving you a lot of time. And so with the next couple of slides, I want to just talk to you about the software and how it works. So VS stands for Villenova Imaging and Analysis Software. It reads that it is a semi-automated image acquisition and processing for quantitative and user-independent data. In short, I can say it's really easy to use. It's packed with automated features for non-expert users so that you can let the software and the hardware do the hard work for you while you're doing something else, doing a different analysis or working on something else. And it's packed with a bunch of advanced image analysis tools, including AI. And once you've acquired your images and you've done the analysis, you can have some real secure storage of your data, knowing that it's stored in a secure SQL database together with all of the metadata. So here is one application of what VS in the ViroTEM system can do. This is called semi-automated multi-level screening. And let me tell you why that is very useful, especially for non-expert users, because it will help you to make impartial and rapid imaging. So when you run a TEM analysis, you put your sample on a grid. In this image, the grid looks huge, but it's actually really, really, really tiny. And you put 3 microliters of your sample on there, you dab it off, and then you put a stain to provide the contrast that you need for your imaging. Sometimes you don't, sometimes you don't put the stain, but most often you have a stain. So you've got your sample on this grid. This is a 400 mesh grid. That means that it's got 400 little squares. And not all of the squares, which is where the imaging takes place, have got good staining that will give you a suitable imaging region or imaging area. Normally you would need an expert user or someone that has got a lot of familiarity with looking at these grids to make the assessment of which area to do the imaging in. We have been able to overcome that by using the semi-automated multi-level screening, where in step 1, we give it an image of a well-stained grid square, and we use that as a reference. Once we have that reference, we can let the system do the hard work itself. We can do something else. So it will go around, it will compare different grid squares and match it against the reference. It will sort them into good grid squares and bad grid squares. And in step 3, it will go back to only the areas that have been deemed to be good grid squares, and it will do the high magnification imaging only in those areas, giving you your step 4, your beautifully analyzed data in the right areas. And this is impartial and it's quick, giving you sort of seamless imaging from sort of just the visual work into your quantitative data output. And once you have set this up, the first time you can save it, and you can just kind of click. So in one click, you can start this beautiful kind of process that starts with your imaging and ends up with like statistics, data, graphs, charts, plots of tens of thousands of particles. And the analysis, finally, in my last part here, is done with 2 different modes of image analysis. And I will go through these. So the first one is called workflows. Workflows are basically highly tunable and connectable modules for tailored image analysis. You basically put different boxes together and you string them together with so you're basically combining different image analysis commands for your image. So here you can see that I really said that really badly, but never mind. Your image analysis takes place here. You're connecting together the boxes, and you can then do detections like identifying particles, AAV particles, intact particles, broken particles, clusters, aggregates, debris. All of that is done in this one image with this one workflow. And that is really useful, extremely useful actually, when you have got something with a well-defined or describable shape. But it can be a bit more challenging to make these, to produce the image analysis and to tell it what you are looking for if what you are looking for changes shapes and structures, like lentiviral vectors, for example. Here we use AI. And this is the final bit here. I'll just tell you a little bit about AI. So AI is a really powerful tool in TEM image analysis. And we work with something called convolutional neural networks. This is a type of AI that is inspired by the biological process in our visual cortex, and it basically uses classified data sets as trainers to distinguish objects in images without the user needing to explain their characteristics. This might not be so necessary if you're having, if you're working with particles with very clear and distinct characteristics, like my wonderful dog here or this cat. And to describe these is not very difficult, but of course to describe the difference between a rotavirus and an AAV, or your pleomorphic particles here, things that change shape, is much more complicated. And this is why it is so useful in the type of analysis that we do. And once you've trained it on a large data set, which we have done at Vironova, you can then continue training it on a much smaller data set to expand its applicability. I'm just going to show you how we do this. So this is AI training for segmentation and classification. You have your TEM image, you perform a set of annotations, and then you run the CNN. It will find the best fit between your input data and your predictive image outcome, giving you your output image and metrics. And the result, here is an image to the left that's been trained. We've given it training on 3 different particles. And then to the right, this is an image that the CNN is seeing for the very first time, and it's doing a really good job, I think, at detecting and identifying these different particles. And you get this AI with the ViroTEM system. It's got 3 pre-trained models, one for AAV, one for lentiviral vectors, and a general one for sort of roundish particles. And so with that, I come to my summary. I hope that I've been able to show you that TEM, that is classically seen as something that is complicated, needs an expert user, and it takes a long time for the image and for the data to be analyzed, can, when combined with AI, actually give you that rapid and routine analytical insights on a whole range of particle features or sample features that you are interested in. And actually, from a sample in a tube to a report, it's only 60 minutes. So you can run at least 8 samples per day and get a full data set for them. That means that we can now go back to those questions I was talking to you about in the beginning, and hopefully we can probably answer quite a lot of them or give you insights to most of these with the ViroTEM system. That means that the ViroTEM system really overall, it just supports much better decisions in the process development throughout the entire, going all the way from upstream to downstream and to administration. So I want you to take home that the ViroTEM system is an intuitive system. It's automated, it's got AI, it's got all types of users, whether you are a non-expert user or an expert user. It's made with focus on viral vectors, vaccines, and drug delivery particles. This is what we know, and this is what we do really, really well. The fact that it's in-house means that you can get your direct analysis of your product quality at your process development or your manufacturing site, which is extremely important. And now as well with COVID, the importance of having things in-house is been highlighted quite a lot, and I think we really understand the value of this. One thing I haven't really talked to you about is scientists don't really want to use a lot of their sample for analysis. You know, it's a precious sample. You've worked hard to produce it, and every microliter counts. So the fact that you only need a really small sample volume to run this analysis is extremely valuable. That gives the scientists, the people the freedom to run this at different stages and of the process development, which is great. And that is the fact that it's a rapid method that only takes an hour from your sample to a report means that it's extremely suitable for routine daily analysis. Finally, giving you a whole range of visual and quantitative data insights and answering many questions with one method, and thereby really saving you time and resources. And with that, I want to say thank you so much for listening, and I'm looking forward to answering your questions. Thank you.
Okay, so thank you everyone for listening. I have noted down the questions, and I'm very happy to run through actually some of them. And again, if there is anything that you wonder, that you think about later, feel free to contact me, drop me an email, let me know, and I'm more than happy to discuss this with you. So let me start by going through some of the questions that came up in the chat. So the first question was if the analysis software is available separately from the TEM. So at the moment, the way that we have our software is in combination with the hardware. So it is for the software, it is the ViroTEM system that you get, and that's where you would have your software package. So it is all packaged in this system. The second question was, I think it was sort of 2 questions or 2 different messages sent around the same topic. The question was about the percentage of region that one can reduce doing good and bad square analysis. And then the user came with a second question, which is, do we sacrifice important regions, clinically significant, while removing the bad grid squares? So this is a really good question. This grid square comparison is actually one that you can fully control. So you take your grid square, the image that you find is this is a grid square with great staining, it's great conditions. You can then say that you want the classification of what it classes as good and as bad. You can give it a certain percentage of how similar it should be to that original grid square image. So if you only want something that is exactly matches the image that you have or the grid square that you've taken, your reference, you can put that value to 100. But you can also put it lower to say 50 or 75. It is up to you. And it's something iterative. You can actually try it out and see how much of the images get binned and how much doesn't. The images are never deleted. They stay there. You can go back and you can revisit those images and you can rerun those images. But we know that, 400 meshes, 400 grid squares, that's a lot of area, and that's a lot of places to go and visit and to image. And some of them will be better and some of them won't be. And we know that classically, scientists or microscopists, they spend a lot of time looking for those good regions. And this is what we're trying to overcome with the smart software. So I hope I've been able to answer that question, which is, you set the limit where you want your images are not deleted. You can always go back and analyze them. So there's nothing lost, nothing clinically significant lost, I would not say. I think there is another question again. So let's see. I see this is right. Okay. So can it be used in understanding the shapes / aggregation of nanoparticles like gold or silver for its use in material science application? We have got some nice examples of these, gold and silver or nanoparticles. This analysis is something actually that Hitachi is really good at and looking at sort of material science application. And this is why we believe that combining it together with our software, we can really do some great analysis and speed some of the things up in these fields and in these regions as well, because you've still got the same powerful hardware. It's just that you've now got a software as well that will help you in your analysis and speed things up. There is another question here that says, I'm also curious about how the automated particle identification data has been validated after identification to be sure that it's identifying particles correctly. Okay, so when you are running your analysis, so I always say that this is a very naked analysis, because you see everything. You see everything that is detected and everything that it hasn't. And you can also tune and work on your workflows or your AI to make sure that you are picking up as many particles as possible. And we are normally in the 90+% of detection. So if we are not getting that kind of detection, we will adjust the workflows or adjust the AI to get that kind of result. So you can always go back and make sure, if you want to, that the particles are identified correctly. Another nice thing that I haven't been able to show you is that when you plot your data, you can look at outliers. You can actually click on individual particles in your graph, and that particle will then be highlighted. It will take you exactly to the image where it comes from, and that image, it will show you which grid square it came from as well. So you will get a direct link to exactly what that detection was. So it will help you as well, even when you're looking at a scatter plot, to really find out what these outliers are and to see if they have been identified correctly or not. There is one more question here. I'm going to read it before I read out loud, but I think, what was the AI part? I missed a little bit. A quick commentary will be appreciated. How about this? We have got quite a lot of material on how we have trained our AI, what we can do, how we go about training it. And please have a look at our website and some of the pop-ups that will come up here as well, because we've got plenty of information on this. And contact me as well. We're happy to send you more content about the work on the AI that we have done. I should also say that we will have a few pop-ups coming up. Actually, 3 of them. One will be a pop-up to request more information on the ViroTEM system. The second one will be if you want to read more, for example, about the AI that we have in the ViroTEM. So to read more about the ViroTEM, we have got a link pop-up coming up as well. And then the third pop-up that we have is a PDF of our ViroTEM data sheet that should have all of the information to you, very easily accessible to just kind of read through and get a good sense of what the ViroTEM system is. And I am also looking forward to talking to you guys, answering your questions. If you want to, I'm happy to align you with our great sales department, and we can always take these questions further. We have got plenty of content. I will answer perhaps one last question that has come up, I think before we wrap up. One is, how is imaging automated? Focus, instrument, and parameter adjustments. So you can, before you start your screening, so this is one way. So you basically set things up and then you press start. Before that, you have got plenty of adjustments that you can make, where you can decide where you want to threshold things, how many images you want to take, what kind of magnification you want to take them in, do you want to use autofocus or not. So you have these options beforehand. It's not really about it just doing everything without you being able to go in and affect or decide how you want that analysis to be done. But that once you have decided how you want your analysis to be run, you can then save that and you can run that over and over again. Or even better, you can get your colleague to run that over and over again without having to go through everything with them. But you say to them, go in that, press start, and you will get the same kind of data with the same sort of parameter settings. I think that we are probably running a bit out of time. I just want to say very quickly that the sample prep is something that I have not spoken about, but I want you to take home that the sample prep is done at room temperature, and it's a very fast method for that. It's something that I have not been able to kind of share here. So with that, I want to say thank you so much for your attention, and I am very happy to have hosted this session. I hope you have all enjoyed it. And again, if you have any questions, feel free to contact us here at Vironova, to go to those pop-ups, or to just email me directly. Thank you very much.